process to convert NAD plus to NADH twice. And these are your products. Two of those, two of those, two of those. Is what mostly you put. Okay. Right. So that takes us. So this is the energetics part. Okay, so I just we can review this stuff. You guys remember what we talked about last time? It was all this energetic stuff, right? So let's break it back down. We just started it with glycolysis, right? So you start with glucose. You convert that to pyruvate. And yeah, and you get 2 ATP, 2 NADH, and you get 2 pyruvate. Then where does it go?
It goes into the mitochondria.
It can. Right, so if it's aerobic respiration, which means it requires oxygen, right? Okay, then it goes into the actual mitochondria. And really, the inside of the mitochondria, the inner membrane, is really like this. The outer membrane is like that. Why do you think it would be like that? Why do you think the inner membrane is all wiggly, but the outer membrane is just flat? Yeah. Exactly. More surface area, right? So technically there's more surface area here. So then you could pack more proteins onto the inner membrane than you could on the outer membrane. And today we'll talk about why that's important. Okay? But this is how it really is, okay, for a mitochondria. Okay? But once the pyruvate, it can get shuttled into the mitochondria where it undergoes what?
Pyruvate oxidation.
That's right. Pyruvate oxidation. Right? Which involves
Carbon dioxide and sodium hydroxide.
That's right. So you take a 3-carbon molecule like pyruvate, it becomes a 2-carbon molecule with a coenzyme A called acetyl-CoA. And I showed you guys what CoA looks like, right? Right? It's just a little like acetate group, a little acetyl group with this giant CoA thing on it. Right? And then, but during that process, it loses a carbon. Right? So that carbon is lost as CO2. And it also what? It gains an NADH, right? Does that sound right to you guys? But that happens twice. So you get 2 CO2 and 2 NADH out of it. And then you end up with 2 acetyl-CoA, right? Times 2. Okay? Because you had 2 pyruvate. Great. So now that you've got acetyl-CoA, what happens to acetyl-CoA?
Citric acid cycle.
That's right. Or the Krebs cycle, or whatever you want to call it. All right? And the very first step of the citric acid cycle involves what?
Being shuttled to the inner membrane.
Nope, not yet. Oxaloacetate. This is acetyl-CoA. Those interact with each other. The CoA is lost. Okay, and it becomes a 6-carbon molecule. Right? Because you guys see that? And
That's good.
there you go. All right, so this becomes a 6-carbon molecule, which is citrate, which is also citric acid. So it's the citric acid cycle. This is citric acid or citrate. Okay. And the enzyme that does that, okay, that takes acetyl-CoA and oxaloacetate, It converts it into citrate. Anyone remember that?
Malate.
Citrate synthase. Then you go through the whole enchilada, right? So we got like the slides over there. Save us some time. You could just look at the slides, right? So citrate goes to, so the acronym that I learned was CIA. Actually, I made this one up myself. Actually, so the original one, I came up with was CIA seriously sucks for making officers. But then I changed that. I was like a 17-year-old. Then CIA stores secrets for making operations. And that's citrate, isocitrate, alpha ketoglutarate, succinyl-CoA, succinate, fumarate, malate, oxaloacetate, which is the one you see here.
Can you go over the succinyl-CoA stuff?
I know. Well, so in this version, it's simpler. So it doesn't have this one, and it doesn't have this one. Yeah. Okay.
What was the first acronym you said?
CIA seriously sucks for making officers. That was my rebel acronym back in the day. All right. I feel like I've grown so much since then. That was like when I was in community college. All right, so do I need to draw this whole thing out? You guys can see it right there. Right? So what are the major products? So you have this whole thing. Okay, yeah, you have alpha ketoglutarate, succinate, fumarate, malate, oxaloacetate, right? And out of this whole thing, the whole point of it is to get what? You get 3 NADs and 1 FAD per turn, right? So you get, and you lose 2 CO2, right? So this molecule right here is a 5-carbon molecule. And this molecule right here is a 4-carbon molecule.
3 NADs and
Yeah, right? So, and then from here on, all the way to oxaloacetate, they're all 4-carbon. So you don't lose any more CO2, but you do lose 2 carbons per turn. Yeah.
So why did you cross out those ones?
Because you don't have to know.
Oh, and then
But they're there.
so like those are being converted each time. So it's being converted.
Yeah, so each of these steps, there's an enzyme that is converting it. And then during that process, every time you lose the CO2, you also get an NADH. Right? So, let's see, I've got. So here you get an NADH. Here you get an NADH. For the fumarate, you get a FAD, a FADH2. Right? So as these reactions are happening, what's happening is redox. Okay, you're getting the reduction of NAD to form these NADHs. Right? You guys remember that whole story? NAD plus to NADH. Oxidation or reduction?
reduction.
Reduction, right? So NAD plus is being reduced. All right, and that is kind of the whole point of this Krebs cycle. You get a little bit of ATP out of this. All right, you get the succinyl-CoA that you don't learn about. That conversion of succinyl-CoA to succinate, you get an ATP. But what you're really getting out of this thing is, and then here, your last one is at the top. You get another NADH. Okay. But if you
Why do
lost,
you get a FADH?
that's just what you get. That's
But why?
why?
Who's that?
That's Anthony.
No, I know. But like, who? Yeah, who?
Anthony, that's Anthony.
He's not in here.
Yeah, he's not here. He's in Zoom.
I know, but like, what's his face?
Oh, he,
What does that matter?
yeah, what does it matter?
Where do you sit usually?
He usually sits here.
Oh,
Yeah, yeah. All right, so this is just this reaction. So the enzyme, okay, that converts succinate to fumarate, okay, it during that reaction, this is just what happens. This is the molecule that gets reduced instead of NAD plus. Yeah.
Okay.
It's FADH.
Mallory, what is the significance of H2O enters? Is on the diagram it says H2O enters? Or is
It's
it
like
just-
it's just
Oh, okay. I thought,
So
it's just utilized by the enzyme.
oh,
whenever we lose CO2, we also gain an ADH.
Yeah,
So
every single time. You notice
For
that? So in
malate.
pyruvate oxidation, here, I should put this in red as well. NAD plus NADH, right?
for malate and oxo
Acetate?
yeah, do we
Yeah.
also lose carbon dioxide?
No, we don't. Because remember, even in glycolysis, we're not losing any carbons, but you still generate NADH. So there's all kinds of reactions that will result in the reduction or the oxidation of NAD.
So when we're getting an ADH, we don't always lose carbon dioxide, but when
That's-
we're losing carbon dioxide, we always get an ADH.
So it would seem. Yeah. Yeah. What's up? Question?
So it goes from 6 carbon to 5, and then the rest are 4.
It goes from 6 to 5, and then to 4.
Yeah.
And then the rest have 4, yeah. All the way up to oxaloacetate, which these 4 join with these 2 to form 6 again, right? And this whole thing happens twice. Okay, so it happens twice, so what you get out of it is 6 NADH, okay, 2 FADH2s, 2 ATP, and 4 CO2.
And this is all in the mitochondria.
Yes, this is all on the inside of the mitochondria. In like the cytosol of the mitochondria.
What is NADH?
NADH is nicotine.
Is it an enzyme?
Oh no, you want to see? It's like a dinucleotide. It's
What is it used for?
nicotinamide adenine dinucleotide or something like that. It's a dinucleotide. So it's like a big. Like, if you want to see a picture of it, this will help you.
I'm wondering more like what it's used for.
What's that?
I'm wondering more of like what it's used for.
I think it's also what is it used for? I mean, it's used for a lot of oxidation reduction reactions. That's for sure. It's a major. So the ratio of NAD plus to NADH is very important to understand the metabolic status of your cells. And this is it. So this is what it looks like. It's like 2 nucleotides. And the only difference between NAD plus and NADH is a single hydrogen. Right? So here, there you go. Here's NAD plus, and here's NADH. Right? Just so you can get an idea of this huge molecule, and that's what's actually happening. So you're like having all these reactions happen in the Krebs cycle so that you can put hydrogens onto that molecule. Right? And but that's what you're really doing is you're moving around electrons. Right? You're loading these things with electrons. What's up?
The 4 CO2, isn't that lost? Because you wrote 6 NADH, 2 FADH2, CTP. Like, I understand that those are all products, but wasn't the 4 CO2 lost?
Yes. You release them.
Okay.
So, but I mean, you generated CO2 during the process. You lost a carbon, and you generated carbon with 2 oxygens, right? Which is a gas which you exhale. Yeah. So when you're losing the carbon, you're losing them, they become CO2. And so it is something that you gain as a result of these reactions.
And then also, where do the ATP come?
So this is 2 CO2 here, right? CO2 times 2. What's that?
Where does the ATP?
The ATP, the ADP gets hydrolyzed. Depends on the textbook. Some say GDP to GTP, but it's basically it's between these steps. There's another step here where alpha-ketoglutarate gets converted to succinyl-CoA, and then it's there. The CoA is lost, and the ATP gets hydrolyzed to become succinyl.
So is this all part of glycolysis, or is
No. So this is glycolysis. Remember, glycolysis is in your cell, like cytosol, if it's us, right? It takes place in your cytosol. If it's a bacteria, glycolysis takes place in their cytosol, right? But everything else, so if you're gonna go this way, the aerobic way, okay, then it's gonna go to the mitochondria. And then it's gonna do all this, and then there's more, okay? Because so far we haven't really generated any ATP. We generated 2 ATP here, and we generated 2 ATP here. That's not very much, right? Why would you go through all that trouble just to generate 4 ATP?
Yeah, that's why I was wondering, like
Why?
we're doing that.
Yeah, exactly. Okay, so here's what we didn't get to last time. Okay, is now,
Don't you get it from the
yeah, exactly. So now what happens is that's all the Krebs, okay? This is the Krebs or the citric acid cycle. Now all of these things are utilized here at the, in oxphos, in the inner membrane of the mitochondria. Okay, this is where the electron transport system, or chain, okay, or it's called ETC or ETS. That's the next part. And the proteins for that carry out that function are located in the inner membrane of the mitochondria. Okay? So here's what that looks like. I'll show you a picture. Okay? This is what it looks like. So you've got these proteins embedded in the inner membrane of your mitochondria. Complex 1, complex 2, coenzyme Q, complex 3, cytochrome C, complex 4, and the ATPase. For this class, we'll just say complex 1, 2, 3, and 4. You got 4 complexes, okay? They're in the membrane, except complex 2 is kind of on the inner side, okay? And what these complex 1, 3, and 4, what their job is to do is to function as a proton pump. Right, so you guys remember the pumps in the channels? These are pumps. They're pumping hydrogen ions, and they're pumping hydrogen into this inner membrane space. Okay, so what that looks like in the grand scheme of things, okay, so we'll say complex 1, 2, 3, 4, and the ATPase. I'll draw this kind of further away to make it easier. Let's just say it's here. Okay. So what happens is, where should I? I'm like limited. Normally I like do it over here.
That was so fast.
You like to pick
That was good.
What's up?
That was good.
What?
Surprised you got through that so fast. I thought that was gonna be like a while.
He has a PhD. He has a PhD.
All right, so here's the deal, is that these guys, what do we do? We reduce them, right? These are all reduced, right? So now what's gonna happen is they're all gonna go and they're gonna become oxidized. Okay, so NADH is going to become NAD+, right? And FADH2 is gonna become FADH again, plus. And then the whole purpose of that, okay, so what we just reduced all those things, now we're gonna oxidize them all. But then what happens is all of those electrons, okay, are going to be passed through this complex. So I don't know if it shows. Yeah, so it kind of shows you here that the electrons from NADH and FADH2, okay, those are kind of donated to these complexes and they're moved across. Okay? So the electrons are being moved across this whole electron transport chain. Okay, electron transport, okay? They're transporting the electrons, okay? But it's that movement of electrons that is what's powering these pumps to pump against the gradient. Okay, so normally, you remember a lot of the other pumps, they use ATP, right? These pumps do not use ATP, they just are powered by the movement of electrons. Okay, but actually they're powered by this whole process where we generated these NADHs and FADH2s so that now they can donate their electrons to these complexes in the inner membrane of the mitochondria. Yeah. How fast is it happening? So what I'm describing to you right now is what you're doing every time you take a breath. Inhale and exhale.
So it takes that long.
I mean, it's happening very quickly.
No, yeah, but
But I mean, yeah. So, because as you inhale, so, and then here's the deal, okay? Then actually, it's probably better to show you here. The final electron acceptor for those electrons is actually oxygen. Right? So you breathe in oxygen, it gets converted to an oxygen radical, which is a super radical oxygen, and it's so radical that it really attracts the electron. So what is actually causing the movement or the pull of these electrons to the entire system is oxygen. It's the electronegativity of the oxygen. Right? Because electronegativity means electrons, right? So this is what's drawing the electrons to it. Right? And so the final electron acceptor of this whole thing is oxygen to form water. Okay?
so with too much oxygen, you like your whole entire body like too electronegative. Is that what it means that you can die from too much oxygen?
I don't know if it's because it makes your body too electronegative, but yeah, you can have oxygen toxicity, yeah.
Play a part in it.
Does the what do you mean play a part?
Too much oxygen, does it like play a part in this? Like, does that work? You can't have too much.
Why can you not have, or why can you have too much oxygen? I don't know. I know it's toxic. Anything's toxic at a high enough concentration. But, I mean, having too much oxygen here isn't going to stop this system. I think there's other reasons why where it becomes toxic at a certain point. I'm not sure. All right, so where were we here? Okay, so the final electron acceptor of all of this, okay, and this is important, so I'll write it here. The final electron acceptor, so the electrons go through the complex, they get passed around through all these proteins. All right, the movement of that powers pumping, okay, of H pluses, so protons, into this inner membrane space. Okay. So now you've generated a gradient, right? Just like we talked about before, a concentration gradient, right? Now you have much more protons inside of here than you do on the inside or on the outside, right? They're all concentrated into that inner membrane space. So by diffusion, they want to go out, right? But they can't because they're charged. Okay, so they can't get out. They can't get out, so they're all there. So that's potential energy. So you went through this whole thing, glucose to pyruvate, pyruvate oxidation, Krebs cycle, all this to give the things to the shuttle so they could just pump a bunch of hydrogens into the inner membrane space. So you did all that just to create a concentration gradient. Right? But these pumps, instead of being powered by ATP, because what you're trying to do is make ATP, Instead of being powered by ATP, they're powered by the movement of the electrons. Okay? And the final electron acceptor, okay, where's my blue? The final electron acceptor of all this is an oxygen radical, which accepts it and then becomes water. Okay, so your product of the movement of all that electron stuff is to interact with oxygen and become water. And now you've just got a bunch of hydrogens in the inner membrane space.
Ready to make water.
No, they're not going to make water. So what you've done is you've generated potential energy, right? Basically, this is really positive. This is not as positive. You got a concentration gradient, right? That's powerful. It's powerful enough to that your body did all this stuff just to do that, right? So that's what you're looking at here. Let's see if I can find a better picture. Blah, blah, blah. I don't know. Oh yeah, because this is the energetics one. So this is the best one we got. Okay. Well, okay. So then, but this shows you everything. So then, here's the deal. So then, after all that, okay, you got all these protons into this inner membrane space, and then what happens is you've got this final enzyme. This is called the ATP synthase. This enzyme right here. Okay, and the job of ATP synthase is it functions as a channel. So it opens and allows the protons to come back out. Okay, that's what you're looking at here. So it opens and allows the protons to come back out. Okay? And during that process of allowing the protons to come back out, it harvests that potential energy. There's like a thing on it that's like a propeller. And it's basically just cranking out ATP. Right? So this thing is rotating. It opens itself up. All these things start flooding out. The hydrogens start flooding out. And that's when finally it takes all that potential energy and converts it to about 30 ATP. 30 to 34. About 32. You know, it's like I don't really care because it's 32. But that's the textbook thing. Because really it depends on your mitochondria, the healthiness of your mitochondria. But you can get up to, let's say up to 32 ATP. Okay? Which is a lot. So finally, Jake, what's up?
So with like the on that ATP, you like depend on like how healthy you are, or like how good your like oxygen level is in your blood.
Yeah. So depending on how healthy your mitochondria is, depending on how healthy you are, depending on how healthy your tissue is, and if you have a disease, or you can have a metabolic disease, you can have different diseases. That's going to affect the overall potential respiration and ATP production of your tissues for different reasons. It could be that you have less mitochondria, or it could be that your mitochondria are not functioning as well. Yeah.
So you said the ATP synthase is a pump
It's a channel, but it's
that just lets
it's a channel because it opens the doors for the protons to get through. But it's also, so by our definition of pumps would put it in other directions, like these guys, right? But it's not just opening the door. It's also churning out. It's working in a way that it's taking that energy and churning out a ton of ATP. That's the name ATP synthase, right? And that's where you finally get all your ATP. You went through all this, and you finally got your ATP.
So from 2 and ADH, we get 32 ATP.
Well, so from a molecule of glucose, you can get, if you go this way aerobically, you can get up to what, 36 ATP, because you got 32 here, you got 2 there, and you got 2 there. Yep.
What's an example question you would ask?
Oh, you missed last lecture.
Yeah.
Functional kind of stuff. I'll be like, a dog comes into your clinic and it's experiencing muscle weakness. It's not capable of doing endurance exercise, something like that. You know, and what enzyme do you think, or what, you know, what, you know? Well, I won't say like what's the name of the enzyme, but I'll be like, do you think is this enzyme that makes, or you know, what do you think is? It will be something having to do with you. You'll have to have a problem and like be able to know this and be able to try to figure out where you think the dog might have a mutation that's affecting its ability or something like that. Or I could ask you something like, well, you know, I got all kinds of questions, and they're all multiple choice.
Can it be no more than 4 choices?
I can't promise anything.
No. A and B, A and C.
Oh yeah, no, I don't like
B and
those.
C.
I don't like those. I don't like those combinations. I don't like, yeah.
What would you say are the most important functional characteristics?
Most important functional parts. Well, if you don't have any of these,
Yeah,
you can't make
say-
ATP. So which one's the most important?
So say if I was studying the night before, like the test, what would be the things I would want?
All of it. All right. Yeah.
So the ATP generated from ATP synthase, does it include the ATP that was used from Krebs cycle?
The ATP. Sorry, one more time.
So the 690 2AFE, that 2 HTP, is that included in
In
the
this?
ATP?
No.
Yeah.
No. So it's that plus that is what you get for every
Oh,
month.
so it's total.
Yeah.
Okay.
So that's is that or this? So you get this from this part. You get 2 from this part, and you get 2 from this part.
Oh,
What is the FADH?
It also gets reduced. It becomes FAD.
Okay, so it also.
It also donates electrons to this stuff. What's that?
So when we're talking about this process, we're both talking about FADH and NADH.
Yes. But you have more NADH than you do
Yeah.
FADH too, from the Krebs cycle.
Uh-huh.
Right? So and then it shows that there, right? So NAD goes to complex 1, FADH2 goes to complex 2. But you know, it's the movement of electrons that powers this, and oxygen is the final electron acceptor to form water. Okay? All right.
Professor,
What's up?
can you see the chat?
Yeah, but I can't. I can listen, but I can't participate in chat while lecturing. But I can answer them later. Okay.
Okay.
All right. So this is if it's aerobic. Okay. I can try to read it. Is it Daniel? Oh, Daniel. Substrate level only occurs in glycosyl. Yeah, I don't know, man. I'll have to answer that later, okay? Okay. So this isn't the only story, right? So that's if you're using oxygen. Remember, in this week's lab, we're looking at fermentation. Fermentation involves, if you're not using oxygen, if you're going anaerobic, right? And if it's us, then pyruvate gets converted to what?
lactic acid.
Lactic acid. Right? And that process, you're obviously gonna lose. Well, it depends what it is, but well, I'll just say you also get. Well, I'm not gonna say anything about the NAD plus and NADH. I'll just say that the lactic acid then gets secreted in the Cori cycle. The lactic acid gets secreted to the liver. Okay, where it can then convert this back into glucose via gluconeogenesis, and then that can then be transported out of the liver and back to whatever cell needs it.
How does the lactic acid again like relate to the
What's that?
explain that or like
Oh, so this is just there's an enzyme. I think it's pyruvate. Pyruvate something. Is it PDH? Pyruvate dehydrogenase? I'm not sure.
what's the little thing like the anaerobic? Is that a number 2 or is that a sign?
Well, that was for this.
Okay.
Because you have 2 pyruvates, so you get 2 lactic acid. If it does. And then we secrete the lactic acid, it ends up in our blood. finds its way to the liver. The liver can convert it back into glucose via gluconeogenesis, but it takes a lot of ATP to do that. And then the glucose can then the glucose is secreted out and can be utilized by different cells. The cells, just like when you eat glucose and it ends up in your blood, you can import it in, right? So this process is known as gluconeogenesis, which is can be utilized, and all that means is when you make new glucose. So you can do gluconeogenesis with lactic acid, with pyruvate, with alanine, which is an amino acid, glycerol, which comes from fats, all kinds of stuff. Should we talk about fats? We'll talk about fats. All right, so that's all mitochondrial stuff. Okay, so then what about yeast? Yeast, they have an extra step. And so you guys saw that in lab. Those of you who have lab today, you'll see that. Yeast don't make it lactic acid, right? They actually convert it to ethanol. And so yeast make ethanol, and that's why we make beer. Or that's how we can make beer. Or wine or whatever. Is through fermentation. By converting the glucose to pyruvate to ethanol. Okay, so, and this is, obviously none of this, it doesn't need the mitochondria for any of this stuff, right? Okay, so I think I will talk a little bit about fats. I do have a slide for it. Let's see, where is it? Yeah, here's what I just said with that. This is what yeast do, right? They convert it into ethanol. You can see that, right? And yeah, this is they use up their NADH in order to do that, which we'll talk about it a lot. Okay. You guys want to try to? So why don't you guys real quick discuss amongst yourselves why some of the characteristics of glycolysis indicate that it is an ancient metabolic pathway.
You want to discuss that?
Yes, amongst yourselves. And if you think you know, you can raise your hand and tell me.
Yo, Anthony, can you hear me?
No way.
What's that?
Yeah, I can hear you.
Oh, cool. My mic's working now. Awesome. Yeah, I was wondering why it wasn't working that entire time. All right.
I can hear you perfectly.
Yeah.
Remember how everything is produced without oxygen, and then it moves to the liver and is converted to glucose?
But not. So this part is called fermentation. But what about this part? From glucose to pyruvate.
What is fermentation?
Because
Pyruvate is lactic acid.
it gets metabolized back to
Wasn't it because mitochondria?
What's that?
Wasn't it because mitochondria was found like in both prokaryotic and eukaryotic cells? So it would kind of like the oldest.
Keep talking amongst yourselves. I haven't heard the right answer yet. I mean, you're kind of right that, but think about a very ancient cell. Do you think it had a liver? No. So then it wouldn't be able to convert that. If it went to lactic acid, or like beer, when the yeast makes the ethanol, it doesn't convert it back to glucose. Right? You need a liver for that. And then for your stuff, prokaryotes don't have mitochondria.
Oh.
So someone tell me why they think that. Yeah, exactly. So the reason why we think that it's the ancient pathway is because you don't need mitochondria for it. So if you didn't have mitochondria, you could still do it. Right? This part. Converting glucose to pyruvate. And even though you don't get a lot of energy out of it, you still get a little bit. Right? You still get some ATP out of it. Right? And so, and it takes place in bacteria and in eukaryotes, right? So it's conserved all the way through. So that's why we think that glycolysis is the ancient metabolic path, because bacteria came before us. In fact, we think mitochondria were bacteria that like we engulfed and then became symbiotic with. Because the aerobic bacteria, like cyanobacteria, they have these same enzymes in their inner membrane in the same ATPase. Okay, so. All right, so one more thing I'm going to tell you about is other stuff. So we talked about glucose. Okay. So what about fat? Let's erase this. So what about fat and protein? What's going on over here? Okay, fat and protein can also be utilized as energy and go into these pathways. Okay, they don't undergo glycolysis because that has to do with the breakdown of sugar. Okay? But for example, fat, you guys remember what a very common fat looks like triglycerides? Remember triglycerides, you got a glycerol group, right? And then you've got those long chains, right? Don't worry, the photosynthesis I'm going to cover today is going to be quick. And then I'm going to drop, it's basically the opposite of the electron transport chain. So we'll finish with photosynthesis at the end. Fat, you make these triglycerides, right? So you have enzymes that separate the glycerol from the fatty acids. Right? The glycerol can end up going to the liver. Oh, that's the long way. It can go to the liver and be converted to glucose via gluconeogenesis. That's what happens to the glycerol if it gets broken down. The fatty acids are converted to what's called acyl-CoA. In fact, I'm going to save you guys because I don't even have room to write it on here. I'm going to save you guys a headache because they don't say it either. All they say is that the fatty acids, I'll tell you this, they bind to carnitine. Have you heard of carnitine? It's a supplement. You could take it as a supplement. The fatty acids, they bind to carnitine. So they become an acyl-CoA carnitine thing. Okay? Carnitine.
Lose fat.
What's that?
Lose fat, right?
It helps. So carnitine shuttles the fatty acids into the mitochondria because you have carnitine transporter. So when the fatty acid binds to carnitine, it could get into the mitochondria. Okay, so that's why it's important. We'll extend our mitochondria here. So once it binds carnitine, it becomes we'll just call it a fatty. We'll call it a fatty carnitine. It's really a acyl-CoA bound to carnitine. But we'll just call it fatty acid or fatty carnitine. All right, and that is able to be then transported into the mitochondria, where it is then converted to acetyl-CoA. So it's a acyl-CoA bound to carnitine. It gets shuttled across, loses carnitine, then it's acyl-CoA. Then that acyl-CoA gets converted into acetyl-CoA. Okay. All you need to know is that the fatty acids bind carnitine. That's how they get into the mitochondria. Then they lose carnitine, and they get converted to acetyl-CoA.
And what happens to the glycerides? They just don't matter.
No, they do matter. The glycerol goes to the liver and can get converted into glucose via gluconeogenesis. It says that there too, right? See, glycerol goes to, kind of wrong. It gets converted to G3P, but then in the liver it'll go to glucose. Or I guess it could also go the other way. Right? Either way. But that's where the glycerol goes. Okay? Fatty acids bind carnitine, go into the mitochondria, get converted to acetyl-CoA. And now you know how important acetyl-CoA is, right? That's like the critical metabolite for the Krebs cycle. Right? So that's what happens with fat. And that's why if you're doing really aerobic exercise, you tend to burn a lot of fat. Okay? And this is aerobic stuff, right? Protein also, we'll just say that, you know, like for example, like when alanine is shuttled, alanine can go to gluconeogenesis. Glutamate can get converted to alpha-ketoglutarate, right? So the proteins end up contributing as metabolites to both gluconeogenesis and the citric acid cycle. I want to ask you anything about proteins. Oh, there is one organ, a very important organ in your body, that prefers to use fat to make acetyl-CoA versus glucose. So if we're talking about skeletal muscle, okay, skeletal muscle loves to use glucose, right? But there's one organ that prefers to use fat to make its acetyl-CoA. And that organ is your heart. Right? Which is pretty important. So the acyl-CoA that I didn't talk about, but you know, the thing that binds to the carnitine, if your heart cannot metabolize acyl-CoA correctly, you can end up with heart failure.
Is that why people who are obese also get heart failure because there's too much for the heart to? Or is it
I think it's more like metabolic disorder. I think it's complicated to understand the effects of like obesity and diabetes on the heart. But certainly it contributes to heart failure.
What is the heart prefer?
The heart prefers to use fat to make its acetyl-CoA rather than glucose. It does both, but it uses fat. It prefers to use fat. Whereas skeletal muscle prefers to use glucose. Okay? All right. All right, one more important thing here. Yeah. Does it talk about all right, so one more thing that's not there that I do want to mention, at least real quick, is there's another important pathway to make glucose, okay? So another way that you can make glucose is from the polysaccharides that you store in your body, right? Do you remember the polysaccharides that we talked about? In plants, what are the major polysaccharides? Cellulose, starch, and what about in us? In our muscle and in our liver, what polysaccharide do we store?
Glycolysis.
Close.
Glycogen.
Glycogen, yes, glycogen. Right, glycogen, right? So we have glycogen, which is these long polymers of glucose that are branching. Okay? So we have those stored in our body, right? And there's a way, so when we're doing exercise and stuff, if we need more glucose, one thing we could do is cleave these off, break them apart, and then that becomes glucose, which we could then go into glycolysis with, right? And that process is called glycogenolysis, which means lysis, or the breaking down of glycogen. Okay? You have glycogen storages in your muscle and your liver. And that's why long distance people like to carb load before events. So they can build up as much glycogen as possible so that they can use it while they're doing whatever it is they're doing. Okay? All right, let's talk about photosynthesis real quick. So I'm switching over to the photosynthesis lecture. Or actually, I'm gonna not, should I even use I'm not even gonna use the photosynthesis lecture. I'm just gonna talk to you the light reaction to photosynthesis. Should I? Should I show you this? Want to see the slide?
It's short.
Doesn't matter.
It's like-
I'll show you a picture. I won't do the whole thing. I'll show you a picture. I have it in my downloads. How you guys feeling about all that stuff? I told you, that's the metabolic biochemistry. Remember I told you you're going to get all the enzymatic reactions you could ever want?
It's like 7 different arrows, 20%.
All right, now let's erase it all. That's metabolic biochemistry. Don't worry, the whole class is not metabolic biochemistry. This is like an introduction to it, just in case this fascinates you and you want to study mitochondria for like I did for like 6 years. Okay. Only 2 years. No, wait. All right, so what you're looking at, and you'll get this in the whole organismal class, right, is the evolution of plants. And I gotta tell you guys that cyanobacteria never gets any credit for being the OG photosynthesis, right? Yeah, they never get any credit. But what do you call that superfood that you guys eat? Kale. No, not kale. What's that? Yeah.
Flaxseeds?
No, it's spirulina. There we go. Spirulina is cyanobacteria. So you're eating ancient bacteria. Gram-negative bacteria. All right. The cyanobacteria were the OG original photosynthesizers. Yeah. All right. So then eventually, we think that they got engulfed by a eukaryote and became an algae. Okay? And there's several different types of algae. All right, you got red algae, you got brown algae, you got green. Brown algae is like the giant kelp. That's a brown algae, right? But we think the one that gave rise to plants was the green algae, right? So green algae is the one that we think gave rise to plants, right? And all of these things are capable of doing photosynthesis. Okay? So what's the difference between an algae and a plant? So this is a prokaryote, but this is eukaryotic. Nope. Also, this is unicellular, although the cyanobacteria do form these kind of like chains, like which are multicellular, and then they have one in the middle that's called a heterocyst. So they're kind of multicellular, but not like these. These are really like multicellular, right? They form like, you know, this you have to see under a microscope. This you can see very, it doesn't look like just one cell, you know? So this is a eukaryotic multicellular, and these of course are multicellular. So what's the major difference? Besides the fact that, well, technically algae is considered a protista, right? Technically these are considered protista, and these are considered plants, all right? That's right. Plants live on land. Unless they're like the water lilies, which developed on land and we think went back into the water. Kind of like the dolphins and the whales. Okay, but plants are on land, algae is in the water. All right. There is some anatomy of a plant. I think you're going to get a lot of this stuff in organismal. But plants have So here, cyanobacteria, we think became just like the mitochondria. We think they were engulfed and became chloroplasts, which are present in algae and plants. Okay? And it's in the chloroplast that photosynthesis takes place. And there's some other stuff. If it's a plant, on the underside of the plant, it has these stomata which open and close to let the CO2 and oxygen, the gas in and out. Right? It's not like us where the gas can just go through.
Professor.
Little channels for the gas. What's so funny?
Calling your name.
Yeah, let me scream.
What's up? Did you call me?
Can you like move the camera to? I can't see like the corner of the board where you just wrote.
Ah. How's that?
Okay, that's better.
All right. So chloroplasts are where photosynthesis takes place. Okay. You don't have to know this stuff, but you know but what you should know is that the chloroplasts also have a very high surface area. So within the chloroplast, there's these organelles that have a really high surface area. Sorry. And those organelles are called, sorry, not organelles. They're inside of the organelle. So whatever. Their name is thylakoid. Okay. So these things, these little folded things that look like discs, these guys are called thylakoids. And it's in the thylakoids, within the chloroplast, where the photosynthesis takes place. Okay? So let me just show you what photosynthesis looks like. Okay? So in photosynthesis, you have a light reaction and you have a dark reaction. Today, we're just going to go over the light reaction. Okay, but what happens, obviously the light reaction involves light. Okay, and yeah, it generates oxygen, which is important, right? And then it also generates a lot of ATP that feeds into the dark reaction called the Calvin cycle, which uses the CO2, okay, to make sugar. But today, we're going to go over the light reaction. Because I don't know if I have time to show you this experiment. I'll tell you real quick that, no, I'll talk about it next time. I'll talk about the experiment next time. But the only thing I want to show you is the actual light reaction, this thing right here, okay? So it looks different. You got light that hits, it excites electrons, water gets split, blah, 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 blah. But let me show you, here we go. Let me show you what it actually looks like, okay? This is what it actually looks like. And I'm going to draw for you my drawing of the comparison between oxphos and the light reaction of photosynthesis, okay? Hopefully you can see this. It starts with a yin and a yang, right? Right? Which side do you think is the light side?
The water one.
Yeah, but I mean, like photosynthesis or oxidative phosphorylation?
Photosynthesis.
Photosynthesis, right? Because it involves light. Right? All right, so photosynthesis is on this side. So that means on this side you have cell respiration. Now, in photosynthesis, let's start with what we know over here. With in oxidative phosphorylation, here's your inner membrane. Outer membrane. Right? Inner membrane space. Okay. You have NADA, NADH is oxidized to NAD plus, and FADH2 is oxidized to FADH, right? Or FAD. During that process, the electrons get passed through all these complexes, and that powers the pumping of hydrogen protons into this inner membrane space, right? Everyone agrees with this? And the final acceptor is oxygen to form 2H, to form water, right? So final electron acceptor is water, starting with NADH and FADH2, which get oxidized, right? Everyone's good on that? And then the purpose of that was to put a bunch of hydrogen protons into the inner membrane space so that they can ultimately get utilized by the ATP synthase to make lots and lots of ATP, right? Everyone agrees with that? Sabrina's like, yeah. Okay. So let's, over here, you got a thylakoid membrane. And you've got a thylakoid space. I always do this in the wrong direction. So that way, thylakoid space starts in the thylakoid space and it goes out of the membrane. Okay, so we'll call this the thylakoid space. Of course, they have to be opposite of each other, right? So here you got, instead of having 4 complexes, you have 2. And there's a pump in the middle, I believe. Okay, cytochrome complex. Now, a key difference, okay? So here you got photosystem 1 and photosystem 2. Photosystem 1 and photosystem 2. Why do you think that photosystem 1 is after photosystem 2?
Because it came first. It came after.
They found this one first, right? So they called that one 1, and then they found this one, and they're like, oh wait, but then okay, scientific, yeah. All right, but then what happens is the light hits, okay, and it gets transduced through a series of steps. But ultimately, what happens is the photons from light excite the electrons on this photopigment in the photosystem 2. Okay, so the electrons become excited. Do you guys know what excited electrons look like? Yeah. They look like this. All right, so it's the same thing though, right? Now you've generated, you've excited the electrons. Now the electrons move, and as they do that, that powers the pumping. Make sure I do this right. Into the thylakoid. Did I do this right? Yeah, yeah. That powers the pumping. I always do this backwards, so if I do it backwards, I'll fix it. That powers the pumping of, no, I see I did it backwards. This way. That powers the pumping of the hydrogen protons into the thylakoid space. Okay, so the protons were here. They get pumped into the inner membrane, or into the thylakoid space, okay? Now here's the deal. Now the electrons that get excited, they're gone now. So something has to replenish those electrons, right? So what do you think does it? Or I'm just gonna tell you. Water does it. So you have water, and it gets split to form oxygen and hydrogen. Oxygen we breathe. Okay, but it's the water that gets split, and then that replenishes the electrons that have become excited. Okay? So the movement of the electrons powers the pumping of the H+ protons into the thylakoid space. This happens twice. So here again, the light hits, right, and it does it again. The electrons become excited. Okay, but this time the final electron acceptor is NADP make sure I write it right. NAD plus, yeah. NADP plus to form NAD pH. Can you see that, Anthony? Yeah, you can see it. Okay, good. You got
You
that?
didn't give me a chance to answer.
You can see it. I can see that you can see it.
Oh yes, I can see it.
Okay. All right, so look carefully here. You start with water, you split it to form oxygen and hydrogen. That replenishes the electrons. The movement of electrons caused the H plus protons to pump into the thylakoid space. All right? Now look carefully. Here you start with water, you end with NADPH, right? Look over here. Here you start with NADH and you finish with water. Right? Yeah. Okay. And then it gets better because what have you done? You put a bunch of hydrogen protons into the thylakoid space. So what do you think is the name of the enzyme that then harvests that potential energy to make ATP? ATP synthase, exactly. All right, so in this membrane is the same thing, the pump. And it's going to utilize the protons from the gradient to then crank out lots and lots of ATP. Just like this, right? So what you're dealing with is like basically they're just opposites of each other. This one uses, and this one oxidizes NADH to form NAD plus, and this one, the final electron acceptor is NAD plus to form NADPH, so it gets reduced. Here, the electrons, the final acceptor is oxygen to form water. Here, the electron donor is water that gets split to form oxygen. You see how these are just basically, but the amazing part about it is that both of them are using that energy to generate these chemical gradients of protons, which are utilized by the same enzyme, ATP synthase, right? The same enzyme, ATP synthase, to generate lots of ATP. Is that crazy? I think it's super cool. Like, so this is like my yin and yang, and hopefully that'll help you remember cellular respiration and photosynthesis. All right. Okay. Oh, so and then this is happening. So in this case, it's the thylakoids. So this is the thylakoid membrane and the thylakoid space. So it gets pumped into the thylakoid, so that So this is like, this is like the thylakoid, basically.
Oh, so there's no like inner and outer membrane. It's just like.
Well, the chloroplast has an inner and outer membrane. The thylakoid doesn't. This is the thylakoid. Whereas here, yeah, it's kind of the.
Is the thylakoid in the chloroplast?
Yes. So the thylakoids are within the chloroplast. This is the thylakoid. In chloroplast.
Versus does the stroma have any effect on photosynthesis or is it oh, it's for the Kelvin cycle, right?
But this is just the light.
The E people, I don't know how to get these years from them.
Let's see.